Sorry, had to start recording. Okay, so hello everyone, I'm Mila Stroganoff, webinar host for the Friends of San Pedro Valley Park, and Adrian Stroganoff is the techie specialist. So with regards to the attendees, if you're a member, we're delighted that you are a member of the Friends of San Pedro Valley Park. If you're not, then perhaps you'll consider joining us and supporting our endeavors. All the information is on our website, and that's friendsofsanpedrovalleypark.org. Our next webinar is on the subject of bats. Corky Quirk will be with us virtually from Yolo County with her little bats. And this is a yearly event for children and adults. So please join us on Saturday, October the 17th at 4 p.m. And then in November, we will hear about spotted and barred owls with Dr. Jack Dumbacker. And he's also from the Academy, and he's agreed to give us a lecture. So we'll be hearing all about owls. So if you have any questions or whatever, please put them in the Q&A section, and we will tackle them at the end of the lecture and have Dr. Luiz answer as many as we can possibly answer without killing them. Oh yes, and don't forget to press the enter button when you write your questions, or if you put something in chat, always press enter, because otherwise it doesn't get transmitted to me or to the speaker. Okay, I think basically that's the main announcements that I've got to make and remembered to make today. And I have the pleasure of introducing Dr. Luiz Rocha, and he is the curator of ichthyology at the California Academy of Sciences. And you've heard us chatting away for the past 20-25 minutes, and you've probably see him on screen. He's a fish expert. He loves his fish. I think all fish, maybe except gobies. I heard there's something about gobies, but he has spent more than 5,000 hours underwater studying fishes and publishing more than 150 peer-reviewed articles and one book. In addition, his work has been featured in many popular media outlets, including CNN, the New York Times, Scientific American, National Geographic, Science Channel, etc., and supported conservation efforts across the globe. So currently his main area of work involves the exploration of little-known deep coral reefs, and that's between 100 and 500 feet depth throughout the tropics. So without further ado, Dr. Luiz Rocha, take it away.
Thank you. Thanks for having me.
Quite welcome.
Can you hear me okay?
Yes.
Okay, perfect.
I'll disappear here.
Okay, and I'll share my screen. Thanks for having me. It's a pleasure, and I apologize again for having to cancel last time. It was just a situation that was totally out of my control, and I couldn't make it. I thought I could make it on time, but at the end of the day, I couldn't make it. Let me turn on my little pointer here. Yeah, I like that thing. I've been at the Academy for 9 years now. And that my main subject of study has always been coral reefs. I've always studied shallow coral reefs, the coral reefs that people know more about. Now I'm studying more and more those deeper, the deeper coral reefs between 100 and 500 feet. And that's most of what I'm going to talk about today. But before I get into the meat of the talk, into the portion of about deep coral reefs, I have to explain to you what shallow coral reefs are, so that everybody's on the same page. So shallow coral reefs, I know everybody has seen pictures, documentaries, videos, etc. Are one of the most beautiful and most diverse ecosystems on Earth, the most diverse ecosystem in the oceans by a long shot. And on land, forests, tropical rainforests are much more diverse than coral reefs. But because coral reefs are the most diverse ecosystem on the oceans, they're often said that they are the coral reefs of, or the rainforests of the ocean. And all of that diversity on coral reefs is basically supported by the structural complexity that is provided by corals. So corals are the building blocks of coral reefs. If you think of a coral reef as a city, corals would be the buildings. And the more corals there are, the more buildings there are. And then the more buildings there are, the more people come in, or in this case, the more fish and the more other animals come in. And then diversity generates diversity. And that's why it's such a very nice, beautiful, big, diverse ecosystem. Now, one thing that's really special about coral reefs is that the base of the food chain in coral reefs comes from this symbiotic relationship between corals and algae. So if you look at that picture there, you will notice that it's really clear water. It's also warm water. So there's a few conditions, a few habitat conditions that corals need to have in order to survive. The water has to be very clear. It has to be warm to a certain point so that they can survive. And the reason why it has to be clear is because corals, they live in this symbiotic relationship with algae, and the algae depends on light to survive, much like plants on land depend on light to survive. They do photosynthesis, and the waste product from the algae becomes the nutrient to the coral. So the algae lives inside the coral skeleton, inside the corals. And then when the algae does photosynthesis, they excrete sugars that are used by the coral as food. And that interchange, and then in turn, corals, when they have their waste products, those are used by the algae as food. So there's this cycle perfectly recycling all of the nutrients inside the coral. And that's what allows coral organisms to survive, to grow. Corals also eat. They also eat plankton. They eat other tiny organisms in the water. But by far, the main source of nutrients for corals is the algae. There's symbiotic algae that lives inside their tissue. So that relationship allows coral to grow and allows the growth and the creation of these very large ecosystems that can be seen from space sometimes, like the Great Barrier Reef. Now, because corals require those conditions that I talked about, clear water, warm temperatures, they are also in the same places where a lot of people are. People also like those places. They like warm temperature, they like clear water. So because they're in the same places as people are, there's conflict, right? People use coral reefs for a number of different reasons. And because people are close to reefs, they damage reefs. They damage it by overfishing, by pollution, in this case, plastic pollution here, by anchoring boats. This is what happens when you add too much nutrients to the water. And by nutrients, I mean sewer. So if there's a sewer discharge close to a coral reef ecosystem, there's always a lot of competition in the coral reef going on between corals and algae, not the algae that lives within the coral, but other types of algae. And those algae that compete with the coral for space, they outcompete, they become better than the corals at growing when there's when there's more nutrients in the water. So the more nutrients there are in the water, the more these algae grow, and that they can get to a point to outgrow the coral. So pollution, sewage pollution especially, on coral reefs is a huge problem because they cause what they call a phase shift. So it shifts from a coral-dominated ecosystem to an algae-dominated ecosystem like this one. So several threats. Most of these threats are mostly local in scope, in geographic scope, and I wouldn't say they're easy to deal with, but there's ways that we can deal with them. Creating marine protected areas, for example, to stop overfishing, or stopping the pollution, to not to stop the algae from outgrowing the coral. But more recently, what's been happening more and more is that in addition to these local threats, we're seeing global threats, especially coral bleaching. So in this case here, the red dots red dots in the, and this is the coast of Australia here. This is a representation of the Great Barrier Reef. So this is almost 2,000 miles in extension. There's a scale back down here in miles. And this is the Great Barrier Reef. It's the largest, the longest coral reef in the world. The red dots are the places where corals bleached. So in 2016, almost half of the corals in the Great Barrier Reef bleached. In 2017, again, there was another bleaching event. And bleaching is a phenomenon that happens. So I just explained to you that corals, they live in this very tight symbiotic relationship with algae, the algae that lives inside their tissues and then gives them nutrients. When the water warms too much, more than what they're used to, when the temperature goes above what they're used to, most corals, they live happily when the water is all the way up to about 82 degrees Fahrenheit or 28 Celsius. If it goes above that by 1 or 2 degrees, the corals, they start to get stressed. And the first thing that happens when they get stressed is that they expel that algae that lives within their tissue. So there's two things that happen that are very damaging to the coral when the algae is expelled. The first one is that they lose nutrition. So as I said, there's a tight recycling relationship between the algae and the coral. The nutrients, the waste from the algae becomes the nutrient to the coral, and vice versa. When you don't have the algae within the tissue, they lose that source of nutrients. And what gives the color to the coral, that brownish or reddish, depending on yellowish, depending on which species of coral it is, what gives that color is the algae. So when they expel the algae, they expel their color basically, and they become white. That's why it's called coral bleaching. And we can see this. So all of these red and green dots here, all of this survey across the entire Great Barrier Reef was done by airplane. So we can see it from an airplane. If the coral reef looks white below the airplane, we know that there's coral bleaching going on there. So all of these surveys were done by airplane, just flying over the Great Barrier Reef. Incredible bleaching at larger scales than we've ever seen before, entirely caused by water temperature, what the optimum temperature for corals is. This is a paper that was published in 2018, so it was reflecting conditions in 2016 and 17, but the bleaching also happened more recently. This article is from April 6, 2020. So this year, there was another massive bleaching event in the Great Barrier Reef. And I kept hearing about, so I grew up in Northeast Brazil, going to coral reefs off my hometown, and I kept hearing about coral bleaching. Back in the 80s and 90s, it was one of those events every 20 or 30 years or so. They were very rare events, and there was a long time between those events, so the corals, they could recover between the events. What's happening now is that the temperature is so high that those events are happening much more often. So there's only 1 or 2 years between those massive events, and the coral doesn't have time to recover. But I kept hearing, I've been diving and I've been a scientist all my adult life. I kept hearing about the massive bleaching event, but I had never witnessed it. And last year, when we did an expedition to French Polynesia, I finally did, which is one of the saddest experiences I've ever had in my life. So this photo here on the left, it's my colleague, the Steinhardt Aquarium Director, Bach Shepard. We're diving here in Tahiti, and this reef looks very healthy. This was in March of, or early April 2019. We were very happy to see that reef, almost 100% coral cover. All the coral was looking really nice. And then we came back to San Francisco. We spent about 4 weeks here. And then we came back to French Polynesia a month later. So in May of 2019, we came back there to do more surveys. And what we found was this. So it's the same location. About 70–75% of the corals were bleached in just that 4 or 5 weeks of time. So that's what it takes. If the water stays 4 to 5 weeks above the threshold of what the corals can survive on, they start expelling the algae. And if the algae stays out of their tissue for long enough, so if they stay white like that, if they stay bleached for more than 3 or 4 weeks, they starve to death and they die. And in this case here in French Polynesia, all of these corals died because of this massive bleaching event, which was something that I didn't plan on seeing, but it was really startling. It's one of those things that I've heard about through my entire scientific academic diving career, but I have never seen it before. And it was really shocking to see it. So let's move on a little deeper now. So all that I've been talking about is about the top 100 feet, 200 feet of coral reef here. That's where almost all of our information about coral reefs comes from. It's from the top 100 to 200 feet. And the reason for that is simple. It's because that's the area you can access using normal scuba diving. So to go deeper than that, you either have to use technical scuba diving or submarines. Now, in my case, I'm a fish expert. So I really don't want to study coral reef fish with a submarine, because trying to study coral reef fish with a submarine, for example, is like trying to study birds on the rainforest with a helicopter. It's really not the tool for the job. When you get close to the reef with a submarine, with all the lights, the noise, all of the fish hide. You can't really study the fish with a submarine. So we opted for technical diving. And because those two options are very expensive, very logistics intensive, submarine is much more expensive than diving, but technical diving also requires a lot of expensive equipment. It requires a lot of training. It requires a lot of time commitment. And there's more than 2 years to get all of the training done. So because there's so much training, so much impediments, and it's very risky, it's very dangerous. It's not a very safe activity at all. Even when you do it following all of the safety protocols that we do, it's relatively safe, but there's a lot of things that can go wrong. So it's not as safe as shallow diving. It's very, very risky. Because it's so hard to get to, very few scientists got there. And this region here, which the official name is Mesophotic coral ecosystem, we nicknamed it Twilight Zone for two reasons. Because it's dark, so the deeper you go in the water column, the less the light penetrates. So basically, the deeper you go, the less light there is and the darker it is. So this area here is, we call it the Twilight Zone, because it kind of feels like you're diving on those twilight hours. And it's also a bit dark, also scientifically dark, because scientists don't have access to it. We don't know what's there. So there's a lot of new undescribed things that we've been finding throughout our expeditions there. So back in the day, 20 or 30 years ago, this is how you'd have to do it with open circuit trimix diving, open circuit mixed gas diving. I don't know how many divers are there in the audience, so I'll kind of keep it the technical diving portion of it as basic and as simple as possible. But suffice to say, you can't breathe air, normal air at depth. The deeper you go, the more toxic, the more narcotic the gases that you breathe become. So nitrogen, which is 70% or 80% of the air, of the gas in the atmosphere that we breathe. Right now, there's a lot of smoke, but mostly nitrogen. It becomes narcotic the deeper you go, and oxygen becomes toxic. It's amazing, right? Oxygen is what we need to survive, but if you get too much of it, it's toxic. So we have to remove those gases and add helium. So we breathe helium at those depths. And when you're doing it with the tanks like this, and then every time you exhale, because we're under pressure, all of the gas molecules, they get much closer to one another, and all of a sudden you need a lot more gas to fill your lung than you do here at the surface. So it's a lot of gas that we're breathing at 500 feet, for example. We can empty one of those normal scuba tanks that lasts for about an hour at 60 feet. At 500 feet, it lasts for a minute. So that's how much gas we need to fill the lungs. And helium is a very, very expensive gas. So it was really impractical to do these kinds of exploration because one, we had to carry all of these tanks, and two, helium is a very expensive gas. At 500 feet, one breath would cost $15, $20 just worth of helium. So we didn't want to do it like that. We want to do it in some sort of more effective way. And the solution we found was this here. So the diver here on the right is using a rebreather. So it's an equipment that recirculates the air as you're breathing, or the gas, the mixed gas that you breathe. And you just keep breathing the same helium over and over again, and then just add the oxygen that you need. And that allows you to stay up to 9 hours underwater, as opposed to this whole paraphernalia here that would be required for the same amount of time. So just to put it very simply, this is how the rebreather works. So it's a closed system. So when you exhale, instead of the bubbles going out like the normal diver, your gas goes back into the system. It circulates back here, it goes back to the back. Then there's a filter that removes the CO2. The CO2 is the byproduct of respiration. Then there's a series of oxygen sensors that add oxygen, but only the amount of oxygen that you need. And then the gas that you're breathing comes back in. So if you notice, there is helium in the mix here, but I don't mention helium through the whole thing. So the helium is nothing is happening to the helium. The same helium you're breathing over and over again. So the helium comes in, CO2 comes out, CO2 is removed, oxygen is added, and the same helium comes back in. So because we're recirculating the helium like this, we're saving hundreds and thousands of dollars every dive just by using this equipment on helium. And the equipment is a lot more compact, which allows us to swim around and catch fish. So with this equipment, we finally have access to those deeper reefs. And there's all kinds of hypotheses we wanted to test. We wanted to scientifically know this ecosystem better. So one of the first things we wanted to study was this. So whenever people talked about these deeper reefs, the first thing they wanted to know was if the deeper reefs were a refuge for the shallow water fauna that's so threatened by all of those local and global impacts, from overfishing to climate change. And for the longest time, for the past 20 or 30 years or so, everybody assumed that yes, that's the case, that the shallow reefs are going to be safe because there's this reservoir on the deep reefs that can reseed them whenever conditions get back to normal or whenever they adapt to the new conditions. And the reason why people believed that was because there's a lot of overlap in species between shallow and deep reef. If you look only at species depth ranges, which I'll show you in a little bit, that's not the right way to look at it. And everybody assumed, without really knowing, but everybody assumed that those deeper reefs were out of reach of humans. And that's an easy one to grasp, right? It's mostly because you can't dive there only with a submarine or technical diving. So everybody assumed that because divers can't get there, nobody can see it, they're out of reach. So we tried to test those two things in the past few years using that technical scuba diving that I just explained. So why is depth range the wrong measure to kind of determine how much overlap there is between shallow and deep reef? So this is one, I'm going to show you two species here, but we looked at hundreds of species, but this is one species called Pseudanthias flavaguttatus. It's the yellow spotted Anthias. It's distributed in the red portions of the map here, so throughout the Coral Triangle and Western Pacific. And the official depth range of this species is 18 to 63 meters. So that's about 60 to 200 feet. So if you think of that, the species depth range is 60 to 200 feet. The way that people used to determine overlap between deep and shallow reef was exclusively by looking at this depth range. So if the species is a species that occurs from 60 to 200 feet, it goes into a column in the database that says that it's a shallow species that uses the deep reef as a refuge, right? So that was assumed to be a shallow species that uses the deep reef as a refuge. And it automatically went into that column, and it was counted at that 70% overlap between shallow and deep there. Now, what we did when we gained the access to those deeper reefs with that rebreather technology and the technical diving and all the training and everything was go down and count the fish using a transect line. We would swim out and then count every fish that we saw. And then we finally had a measure of actually where this species is. And when we looked at it, it was very different. 99.8% of all the individuals of this species that we saw were between 50 and 60 meters depth. So that's between 170 and 200 feet depth. And the others were deeper than that. So what's the difference here between 18 to 63 and 50 to 60? The difference is that this depth range, it includes every single record through history from where that species was collected or seen. So this species was described in 1980. It was described from Japan, way up here. And in Japan, the water's colder because it's way up north, right? So when the water's colder, something very interesting that happens to those deeper loving species is that they go shallower. They follow the temperature contour. So in a few locations, this species is very widely distributed through the tropics, but in a few locations in Japan, because the water is so cold, they go up shallow. So on those locations, that's where it was recorded at 18 meters. But in the vast majority of the places where it occurs, it doesn't occur at 18 meters. So not only this species is not a shallow species that uses deep reefs, it's not even a shallow species at all. It's a species that is almost entirely restricted to deep reefs, and it only goes up to shallow reefs when the water is really cold outside the tropics. So there was a lot of bias in that estimate, that very high estimate of species distributions. And we saw that over and over again for different regions. This is an Atlantic species, Comeris ancrisura. It's distributed all the way to from Florida to Rio in Brazil. And again, throughout most of its range, it's only a deep species. You never see it shallower than 200 or 300 feet. But when you get to South Brazil, to Rio and Sao Paulo, it's at 10, 20 feet because it's cold water there and they come up with the cold water. They like cold water, they come up with the cold water. So again, another deep species that doesn't occur shallow, and it was counted on those databases as being a shallow species that can use the deep as a refuge. So a very, very wrong way of looking at that. Same for the corals, which are the ones that are suffering more directly with global climate change and global warming because they bleach. So those two species here are the most threatened species of coral in the world. There are two Caribbean species, and on the left here is the staghorn coral, and on the right, the elkhorn coral. Their populations declined by 95 to 98% throughout the Caribbean for a number of different reasons. Right now, the main threat is, again, climate change, just like what's happening throughout the world to other coral reefs. And their official depth range is 1 to 60 meters. But the vast majority of the individuals we find are from 0 to 10 meters. So that's surface to about 200 feet, it's the depth range, but the actual individuals are the surface to about 30 feet. So they're really an only shallow species, and in very rare occasions, someone saw one deep one, but there's no way that that one deep coral could recede all of the populations from the shallow reefs. There's no viable population in the deep reefs. So to study this in a lot more detail, instead of looking at those depth ranges, which include all of those outliers, we actually went to the locations, we counted the fish, we photographed the coral, and we saw what was actually there. Instead of looking at database level, global scale, depth ranges, we actually went to the reef and we saw what was on the reef. And the results we got were startlingly different from everywhere else we've seen before. And that included a myriad of new species. So this is just a few examples of the new species, which is one of the most exciting things that comes from my work, I think, is seeing all of these new species for the first time. It's very, very exciting when you get to a place that nobody ever dove before, and you put all of that gear on. It's about an hour or two of preparation just to get all of the gear ready to go, and then you finally go and dive, and you don't even know if there's going to be a reef in there or not. And when you get down, invariably you find one of those beautiful new species. On average, we find 3 to 4 new species per expedition we do. These are just a few examples. Those two in the left here are the two most recent ones that we named from French Polynesia. So from that trip that we did last year where we saw all of the bleaching. Here, just to give you a summary of new species that we found recently, and that's a really funny graph or table, I think. Those are new species. That's the expedition here in Fiji. This is mostly run, so the yellow expeditions here are mostly run by colleagues of ours. The white expeditions are the ones that our team did. That's the number of new species. That's the exploration time per hour. So that's the number, that's the amount of time that we spent at those depths, at the mesophotic depths. So when you're diving deep, you can't stay very long because there's a lot of gas that gets dissolved into your tissue. And to safely remove that gas from the tissues, we have to go up very, very slowly. So if we spend about 10 or 15 minutes at 400 feet, it takes us 5 hours to come back up. And the longer we stay down, the more gases get dissolved into this tissue, so the longer it takes to come up. So there's very little time that we're allowed to stay at those depths. So we only stay 1 or 10 or 15 minutes in a dive that can be 5, 6, 7 hours long. That's why the numbers, the exploration time here is so small. So in 14 hours of exploration, the Philippines, we found 14 species in the Philippines, spent 8 hours of exploration. That comes up to, here you go, the punchline number, the number of new species per hour of exploration. And that's just fish. So that's how much we don't know about these ecosystems. We're finding anywhere between 2 to 11 new species of fish per hour of exploration at those depths. So in addition to them being very distinct, as I just showed, from the community level, there's also a whole lot of new species that we don't even have names for at those depths. There's also a lot of new records. So these are all only exclusively deep species that were described from one location, and we found them in another location 1,000, 2,000 miles away, mostly because we don't know anything about the distribution of those species, the geographic distribution of those species. If they were described from Indonesia and we go and we find them in the Philippines, it's not because they've never been there and got there recently, but because nobody dove in the Philippines before to those depths. So we're the first ones, and we're kind of only now starting to understand the distribution of those species. Some of them are like completely spectacular looking, like this Odontantha scatayama that I photographed at about 480 feet depth in the Marshall Islands. But the colors of those fish are just incredible and mind-blowing. This slide stopped here. Okay, there you go. So this is the average summarized result of all of those transects that I did. We did hundreds of transects. We counted a lot of fish to determine how much dissimilarity there was between the shallow and the deep reefs. So if you remember from one of my first slides, I said that using the depth ranges, which was the wrong way to do it, there was about an estimated 65 to 70% overlap between shallow and deep reef. When we use the transects, this is not overlap, this is dissimilarity. So it's a measure, an ecological measure of community comparison that's called dissimilarity. So this is how different the communities are between shallow and mesophotic. So anywhere between 87 to 99.7% different. So there is no way that these reefs can be a refuge to the shallow, the deep reefs can be a refuge to the shallow reefs, simply because the set of species that there are in the deep reefs are completely different. There's a lot of new species that we don't even know yet, we don't have names for yet, and there's a lot of species that only occur in the deep reef, but we thought occurred in the shallow, but they don't. So very, very different communities. Now to that second assumption, right? Are they really out of reach? So that's one thing that we didn't initially plan on doing, which was looking at human impact at those depths, but we couldn't avoid doing it because we saw human impacts everywhere. This is a trip we did to the Bahamas. It was just a few days after a hurricane passed through the Bahamas. And hurricanes were one of those things that was originally believed that it only affected shallow reefs. So when a hurricane passes over a coral reef, it creates a lot of waves, a lot of current, and obviously, I mean, there's a lot of corals that break and die and then get flipped over, coral heads get turned around. So there's a lot of destruction in the shallow reef. But everybody thought that because the deeper reefs, so they're deeper, there's no wave action down at 300, 400 feet. Everybody assumed that they were out of reach from there was no impacts of hurricanes on those deeper reefs, right? So this is a picture I took at about 450 feet in the Bahamas, 2 days after a hurricane had passed. And normally this reef wouldn't look white like this. All this white here is sand that basically was steered up from the shallow reef. This got all steered because of the turbulence caused by the waves and the currents. And that sand rained down on the deep reef, and it smothered, it covered all kinds of invertebrates that live here. All the corals, this coral has a little tip sticking out here, but it's much bigger colony that was completely buried by sand. So the impacts are there, even from events that we thought would impact them. Natural events like hurricanes. There's a lot of coral bleaching. Everybody assumed there was no bleaching. This colony here is bleached. Everybody assumed there was no bleaching at Mesophotic coral ecosystems, at twilight zone reefs, at deep reefs, because the water is colder there. So the two things that you notice when you go down diving at those reefs, one is that it gets darker the deeper you go, and second, it gets colder the deeper you go. So the absolute temperature in the water is colder at those deeper reefs. But for the corals, what matters is not the absolute temperature. So the shallow corals are used to 82 degrees year round. If the temperature goes up to 84, they bleach and die. The deep ones are used to 60 degrees year round. If the temperature goes up to 62, they bleach and die. So it's the variation in temperature that hurts the coral. It's not the absolute number there. So there's bleaching all the way down. There's coral disease. There's all kinds of impacts we saw all the way down. This is a picture that kind of summarizes it. This is a sea urchin, one of those decorator sea urchins that picks up things that are around it to camouflage itself with. And this is a fishing line. So the sea urchin is attached to a fishing line. This is at about 450 feet depth in the Philippines. And this blue thing here is a piece of plastic. So the sea urchin was camouflaging itself with a piece of plastic, which was working very well in this case because there's plastic everywhere in the bottom. And that was attached to a fishing line. The Mesophotic, the deep reef, is very close to shore in the Philippines, so a lot of people fish there. There was a lot of fishing line, no big fish to be seen. So even the impacts of overfishing are all the way down there. This is the slope of a reef in Palau. This is an anchor line from a boat. There's fishing line on the reef here that you can't really see because it's a wide angle picture, but the impacts are obvious everywhere we went. And nowhere they were more obvious than it was in Guam. So Guam is the most impacted island in the entire Pacific. There's a lot of human impacts there. There's a very high population. It's a densely populated island. There was a lot of impacts during the war. It was seen for a lot of the scene of a lot of battles during World War II. So it was bombed a lot. There's a large U.S. military base there. There's a lot of impacts to the reef. The shallow reefs are in really, really bad conditions. But the deep reefs are in relatively okay conditions. So they escaped some of those impacts that damaged the shallow reefs a lot, but they didn't recede. They didn't function as a refuge for the shallow reefs, because the shallow reefs, so these are the shallow reefs from Guam here, they are severely impacted and not recovering. Mostly because the deep reefs, even though they're in much better shape than the shallow reefs in Guam, the species from there, they can't survive in the shallows. They're typically deep species. They're doing their thing in the deep, so they're not going to move up shallow. So in summary, not a refuge. So I'm going to tell a story of an expedition we did to St. Paul's Rocks in Brazil that kind of puts everything together. It summarizes everything that I just said. This is an island that's about 1,000 kilometers. That's what, 700 miles from shore, from Brazil. 1,000 miles, 1,500 miles from Africa. So right in the middle of the Atlantic, in the middle of nowhere, one of the most isolated islands in the world. This building here is the only building in the island. And the only reason why this building is there is because the island, the territory, the island belongs to Brazil. And in order for Brazil to claim the exclusive economic zone around this island, they have to keep a permanent presence in the island. So the way they solved that, the Navy, the Brazilian Navy, built this research station there, and they keep biologists there that don't charge, right? So that's the cheapest way to keep a permanent presence in an island anywhere in the world. It's by sending biologists to stay there for free. So the biologists go and stay for free in this research station, and the Navy goes there once every few months or so to maintain the station, clean the solar panels. This is all bird poop on top of the solar panels here. And it's a tiny, tiny island. So this picture I took looking into the research station. If I turned around and took the picture looking the other way, all you would see would be ocean. So that's how small the island is. We went there aboard the the Alucia. It's this big ship that supports ocean exploration. There's also a submarine on board, and all kinds of support for our deep diving. And we use the platform because we couldn't do it from that little research station there. We did our dives there. We did our transects. So that's Hudson, one of our postdocs, counting fish. That's the little slate that he has, and he counts the fish and writes down all the fish he sees. This is Maridius Bell, our dive safety officer, who comes with us in every expedition to make sure that everything is conducted in the safest possible way. And we were stunned by some of the things we saw. So here's a video that I want to share. And if you have your audio turned down, I mean, you're hearing me, but turn it up a little bit more so you can hear the sound from the video too. Hopefully it plays there. So I'm going to pause it here. This is the dive computer that's connected to that rebreather system that I explained in the beginning. That's the depth there, 419 feet. When we have 83 minutes to end the dive. So we have 83 minutes of the decompression to spend decompressing at that time. And Maridius was filming, that's me down at the bottom there. Right here, that's me. So I'm kind of catching a fish there, and then Maridius see the shark. You hear him? So the voice, the chipmunk voice, is because of the helium that we're breathing. And he's trying to catch my attention. He's saying, Luiz, look at the shark. And I'm like, no, leave me, I'm catching a fish. So Hudson's right here. He's like, Hudson, look at the shark. Hudson, look at the shark. And Hudson's saying, okay, I'm catching a fish. So I'm gonna pause it here again. I don't know why my pointer is not working, but if you look at, there's under the fin here, under the pectoral fin of the shark, there's a line. You see that line there? It's a fishing line. So that species of shark is a super rare species. It's only found at those depths. That probably is a fish that nobody has ever seen before, an individual fish that nobody has ever seen before. And it has a fishing line attached to it. And that's in the middle of the Atlantic, in a place, in a tiny, very isolated island, in a place where there's only one building with 4 people in it. I'm gonna roll the rest of the video here so you can see the excitement. You see the fishing line there attached to the shark mouth. So the funny thing about this is that neither, there's Hudson and I again there. The funniest thing is that neither Hudson or I saw this shark. The only person that saw the shark was Maridius, was the guy that was filming. And the reason why we didn't see it was this, and how stunningly beautiful this fish is. This is a fish that we saw for the first time on that island, and we described it as a new species. We named it Tosenitis Aphrodite because it was so pretty that it hypnotized us and made us not even look at that shark. So we made it after the Aphrodite, the Greek goddess, right? Logically. One of the most spectacular discoveries we've ever done, and something that I'm never gonna forget. So we also saw in that island, tiny island in the middle of the Atlantic, with one building, 4 people, lots and lots of signs of human impact: trash, pipes, pieces of tissue and human clothing, and fishing line, anchor lines. So we saw impacts at Mesophotic reefs 1,000 miles from shore, in a place where with only one building, one of the most isolated islands in the world. So those reefs, not only they're very different, but they are also impacted by many, many things, almost as much as shallow reefs. They're not in as bad shape as shallow reefs are, because the main building blocks of that ecosystem are not corals that have algae inside their tissue. So bleaching, even though it happens at those depths, it's not affecting the entire ecosystem the same way it's affecting shallow reefs. But they are impacted by overfishing, they are impacted by pollution, they are impacted by sedimentation, they're impacted by a number of different things. So they're very different ecosystem, and they are heavily impacted, which completely overturned the more accepted idea that they were intact refuges that could eventually function as a seeding bank for shallow reefs whenever shallow reefs really start dying. So that brings us to this topic of mesophotic or twilight zone conservation, which is something very hard to do. If you think of a shallow reef, like in this picture here, it's not completely alien to people. When you say we have to protect the coral reefs, people usually agree because it's a very beautiful ecosystem. Everybody has access to it. You can go snorkeling, even if you can't go diving in it, you can go snorkeling and see it. But deep reefs are very different. If you think about the typical mesophotic ecosystem, that's what it looks like to most people. We don't even know where they are. In a lot of places, we don't know if there are mesophotic ecosystems and how preserved or how impacted they are. So it's hard to convince governments to protect those reefs because we don't know anything about them. So, and people protect what they love, right? So one of the things we do at the Academy is trying to bring attention to those reefs at the California Academy of Sciences to inspire conservation of them. And that's something we do constantly. We do it through our public floor. The Academy has an entire wing of the aquarium. It's called the Twilight Zone exhibit. And it's dedicated entirely to those reefs. All of the organisms, all of the corals, all of the fish you see in those tanks in the Twilight Zone exhibit are from those depths. It's a spectacular exhibit. I recommend whenever the Academy opens again, I recommend you go back there and take a look at it. There's even one tank where we keep only species that don't have names yet. So we go on expedition, we bring those new species, and sometimes we bring them alive, and then we put them in this new discoveries tank that only has new species, something really, really cool and unique. There's no other aquarium in the world that does this. The city of San Francisco just announced that they're allowing indoor museums to open on September 21st. So hopefully we will open shortly after that, maybe a few days after that. I don't think we're going to be able to open on the 21st, but after the 21st, shortly for sure. So looking forward to that. Something else that we do to bring attention to those beautiful, spectacular discoveries is share our findings with the media. The media is really keen on talking about these kinds of things, and they're really interested in it. I mean, really big media outlets, Fox News and CNN and all kinds of cool magazines and outlets talk to us about this. And that Tosinoides Aphrodite, the Greek goddess fish that I talked to you about, it's funny that about half of the press releases that came out about it focused on how beautiful the fish was, and then the other half focused on the whole shark thing, or the video of the shark. They shared the video. It was really, really cool to see. And those things, they bring attention to the plight of those reefs, because then we can go back to the government and say, look, this fish is all over the media. Everybody is interested in it, and you're not protecting it. So with this particular fish, we were able to successfully lobby the government in Brazil to protect most of the area where this fish occurs, in that super isolated island in the middle of the Atlantic. So hopefully some of those impacts that I talked about are going to be less in the future now that it's better protected. This one is another one that we had a lot of fun with. We named it Cirrulabrus Wakanda, over the mythical Wakanda nation from the movie Black Panther. And because the name was about the movie, and again, the media loved it and was a media frenzy, and there was all kinds of articles about it. This one came out on CNN. So really cool stuff that kind of brings the attention to the existence of those deep reefs. And that was about all I had to say. I mean, the team that helps me do this is giant. I couldn't do this by myself. There's a lot of students and postdocs and colleagues from other institutions. We collaborate a lot with colleagues from the University of Hawaii and the Bishop Museum in Hawaii. We do a lot of things together with colleagues from the Smithsonian. So with that, I'll stop sharing here and open for questions.
So let me check what we have in the question section. Well, have you found treasure in the ocean before?
Ah,
Julie is asking about treasure.
treasure.
Mm-hmm.
Once in a previous life, so before becoming a biologist, even I've always been drawn to the ocean, and I always dove and I always went diving, and I, I always liked diving in shipwrecks, and I found all kinds of cool things in shipwrecks that had never been explored before, like old porcelain and bottles and things like that. I mean, more artifacts, but not really treasure.
Okay, and then Bonnie is asking, short exploration time, you had big numbers of discovery, more a comment. So you have to be exceptionally quick with regards to selecting fish. I mean, to know what is already been discovered at those depths and what hasn't. It's like you have to have this photographic memory.
Yeah,
And
yeah.
in very short time.
Right. And yeah, I mean, we're constantly studying those fish. So for us, it kind of comes relatively naturally when we see something in certain groups of fish. So, I mean, you mentioned the gobies at the beginning. That's one of the reasons actually why I don't like the gobies, because it's there's more than 1,000 species of gobies out there. So I can't tell if it's a new species of goby or not. I have to send it to a specialist. So my deal with the gobies is that it's not a very charismatic group. It's not like an angelfish or an antias or a big colorful fish like the ones I just showed. So there's not a lot of scientists studying them. And that the funnest and more effective way that I found on social media, especially, to bring attention to the plight of the gobies, to kind of bring more people to study gobies, was trashing them. When I say they're nice, people are like, yeah, they're nice. But when I say, oh, gobies are nasty, and then people, no, they're not, they start defending them. And then all of a sudden my student says, oh, I want to study gobies. I'm like, yeah, nice. So it's reverse psychology.
It's working, obviously.
It is, yeah.
And when you were mentioning temperatures, you were saying 1 or 2 degrees, basically. And are you discussing 2 degrees Fahrenheit or centigrade?
It depends on the location. In some places, when the more stable the temperature is, the less variation is needed for the corals to stress out and bleach. So if it's a place where it's constantly year-round 82 Fahrenheit and it never changes, and there are places like that, when it goes to 83, 84, the corals bleach.
Really?
Yeah, but then there's places, so that would be places like in the equatorial Pacific, so maybe Fiji and Vanuatu and Marshall Islands, is places where the temperature is really, really constant. In other places, like in the Red Sea, it fluctuates widely. So in the winter, it can be 76, 78, and then in the summer, it goes to 85, 86. In those places, because it varies a lot naturally, the corals there are more used to the variation. So there's a bigger, no, they only start to bleach when it gets to 88 or 90. So when it's 4 or 5 degrees above.
So they've evolved with that
yeah,
dynamic change in temperature.
Right. But they're also bleaching because even there, the maximum is going way above what they tolerate.
Oh, heavens. And is it true that the hooks caught in fish actually dissolve in a fish's mouth? Is that true, or is that a
they
tale?
dissolve. It takes a long time, but they do dissolve. And they're making better ones with a different metal type that dissolves even faster, but it takes years to dissolve. So it does dissolve, but it takes years. It takes a while.
So let me check and see. So how are new species names determined? For instance, this goes back to this whole question Sharon is asking about naming.
Mm-hmm. It's an interesting process. So the person that first finds that species and determines that it's new, it's the one that gives it the name. Usually there's always two components to the name. When I talked about the one that we named after the Greek goddess Aphrodite, the full name of the species is Tosonoides aphrodite. So Tosonoides is the genus, The species is both the genus and species. Centosanoides aphrodite is the species. The genus name was already there. So the genus is a larger group with very closely related species. And then the species name, because we discovered it, we chose that name because of the circumstances of the discovery. So sometimes I name it because of a distinctive character. That one of the species I named was Himilon squamipina. And squamipina means scales on the fin. And we put it that name because it was the only species in the genus Himilon that had scales in the fin. So it's a Latinized version. It's squamipina in Latin means scales on the fin. It has to be Latin or Greekish kind of, and you can name it after people too. I named one after my wife once, so Helicaris Claudia. So, so in short, the person that discovers it is the one that gets to name it. And to officially name it, we have to write a very detailed morphological description, and then we have to submit it to a scientific journal. And then the journal sends it to peer review. So they send it to other scientists that also describe species, so they can review the manuscript and make sure that we're saying is a new species is actually a new species. Then it gets accepted in the journal, and then when it's published in the journal, it's recognized and it gets that name. And theoretically, that name stays for eternity. So some of the species that I showed in this talk, they were discovered and described in the 1800s.
And how much, this is basically taxonomy.
Yeah.
And how much is molecular, is the newer technology in DNA and molecular biology, is that being applied more in terms of naming fish?
Yes, we use it all the time. It was one of the main things that we use, actually. So traditionally, normally, what we would have to do is count morphological characters on the fish. So say count how many dorsal fin spines there are, how many pectoral fin rays, how many caudal fin rays, take an X-ray and see how many vertebrae the fish has. And then we would take all of those counts and then measure the fish, see how deep the body is, how long it is, measure, and then calculate all the body proportions, how big the head is in relation to the rest of the body. And then compare all of those morphological proportions to all the morphological proportions of all of the other species in the genus. So all of the other closely related species. Now we still have to do all of that, but we only do it after we do the DNA test. To make sure it's actually new. So when we do a DNA sequencing and we compare it to all of the available sequences of the closely related species, and we see that it's 3 or 4 or 5% apart, which is, for example, how far we are from chimps. We're about 5% different from chimpanzees. Then we say, yes, this is a new species. And then we go back and then we do all of the boring morphological work. But we don't have to do all of the detailed comparisons because we already know that it's a new species. And then we use, in the description, we use both the genetics and the morphological side of it. But the morphology is still essential because genetics is not available to everybody, right? So it might be a scientist from another country that finds a similar species, but it doesn't have access to genetics. They can still do the morphological work to determine if what they found is the species that we described. Or
okay, you froze. All right.
not see you.
Okay, so yeah.
We're back. Okay.
Yeah, you're back. So it says here, Jim Steele is asking, some bleaching happens from cooler temperatures. Variations in upwelling of cooler temperatures can cause bleaching at depth or prevent bleaching in shallow. Have you seen anything regarding the effects of upwelling?
Yes. So one of the places that we visit a lot, as part of our field work, we go there multiple times a year sometimes, is the Philippines. And the area we go to in the Philippines is an area that is geographically very complex. There's a lot of islands, a lot of seamounts, and it's between the Pacific Ocean and the South China Sea or the Philippine Sea. there's strong currents, and those strong currents, they cause the upwelling that he's talking about there. So upwelling is when cold water from the bottom comes up to the surface.
Mm-hmm.
And it brings cold temperatures. So in places like that, there's a lot less bleaching in general, because the pool of water, even when the surrounding water is very warm, because there's this cold water coming up, the temperature is kept low. So in those areas is where we saw the healthiest coral reefs because of the upwelling.
Oh.
Cold water does kill the coral too, but it's a lot more rare for that to happen because it doesn't stay cold for too long. So when it's a warming event, the water can stay warm for a month, 2 months, 3 months at a time. When it's an upwelling event, usually it comes in and out within a few days. So if the stress is that short, the coral can recover after. But when the stress is very long, that's when they die. Yeah, but the areas where there is upwelling are some of the areas that we hope are possibly going to naturally survive to all of these crazy times that we're going through.
So I have a question here in regards to rehabilitating coral reefs, the efforts being made, and whether can reseeding be successful if water temperatures remain elevated?
Right. There's a lot of research being done in that area, including by us at the Academy. The answer to the second part of the question, if they can survive, if the temperatures can stay elevated, is basically we don't know. We haven't enough data to say one way or another. We hope yes they can, because by reseeding them, by multiplying their numbers, hopefully we're multiplying their genetic variability. And with more genetic variability, there's a higher chance that 1 or 2 are going to survive, and the ones that survive will hopefully be adapted to those new temperatures. Yeah. But we don't know. What we do know is that it helps some species reproduce. For example, those species in the Caribbean, the 2 very threatened ones that I talked about in my talk, the 2 croppers, the elkhorn and staghorn corals in the Caribbean, there's so few of them, they're so far apart from each other, the surviving colonies, that they can't successfully breed anymore. The corals don't swim, they're attached to the bottom. So when they release their gametes, their sperm and eggs, they have to be close to each other to be able to meet and fertilize. Mm-hmm. And the few surviving colonies are so far from one another, they're not even reproducing anymore. So one of the things that we're doing is catching those eggs and sperm, putting them together in the lab, and then putting the babies back in between the other colonies, so that the babies can become eventually adults and then connect the separated ones. So it helps to a point. We're hoping that the help that we give in them through these reseeding or restoration projects, we're hoping that it can buy corals some more time. But the ultimate solution is bringing temperatures back to somewhat to normal.
Yeah, that's a major endeavor.
Mm-hmm.
Okay, let's see. Okay. Yes, Bonnie, this particular lecture is recorded and will be available on the website. And there's another question from Sharon about, are fish more or less colorful at depth?
Yeah, so the next question is about color too, from Pat and Jim Creamer. So I think I'm going to try to respond both at the same time. Mm-hmm. Because one answer has to do with the other. They are, in general, they are more colorful. And they are more red and more pink and more yellow, all warm colors. And it makes no sense, because as I said, the deeper you go in the water column, the more water light is absorbed. So light is taken out of the water column, is getting reflected back. And at those depths, at 400, 500 feet, the only wavelength that penetrates that deep is blue. So the only light that we see at those depths is blue, because that's why, one of the reasons why the ocean looks blue. It's because that's the light that gets reflected the deepest in the ocean. So there is no red light, there's no yellow light. So basically, why do they have those colors if they can't even see them? So we're actively trying to answer those questions. And one of the most accepted hypotheses, one of the ideas that we have of why that happens is because those colors make them invisible at those depths.
Invisible.
Yeah. So red looks completely black if there's no red light to reflect. So it's basically having a color that it doesn't reflect. So they don't call any attention. It's not like in the shallow reef, everything is colorful. So being colorful almost makes you camouflage. But in the deep reef, everything is blue. So if you're a blue, you stand out. But if you're red and there's no red light, then you're almost invisible. So they use it for almost for camouflage. And it's bright for us when you put an artificial light on top of it. Mm-hmm. But it's almost invisible at depth because there's those wavelengths don't penetrate that deep.
Hmm. And what about the actual, Margaret Goodale is asking about the how fish adapt from these depths when you bring them up.
So if you go to the exhibit at the Academy, there's a whole section there dedicated to exactly this topic and how they survive at the aquaria at the Academy. And we solved that. So normally, if you went deep sea fishing anytime, or even normal fishing sometimes, depending on how deep you're fishing, when you reel a fish up, their swim bladder expands because the pressure decreases, and then it kind of comes out, and then the eyes come out. Then basically the fish explodes. To avoid that, back in the day, when people caught fish for aquaria, they use a needle to puncture a hole in the fish swim bladder, which is basically an airbag, and then let that vent that gas out as the fish comes up. Mm-hmm. But we at the Academy, our team there, the aquarium biologists, they invented this decompression chamber for the fish. So it's a chamber that we put the fish inside, and then we seal the pressure. Then we leave that small chamber pressurized, and then we slowly, very slowly, over days, we remove the pressure. So if the fish is given days to adapt, then the swim bladder very slowly releases the gas without stressing the fish. It's much better than puncturing it with a needle. There's no risk of infection. And after they go through that process in the decompression chamber, they survive fine at the ambient pressure, normal, just 1 atmosphere. That's the pressure that we are here. Mm-hmm. What we do have to do in the aquarium is keep the light level of the tanks low, yes, and the temperature low. So the aquarium, if you walk around before you get to the Twilight Zone exhibit at the Academy, you go through the shallow reef exhibit. And the shallow reef is all very lit, very bright, lots of light. And then when you turn the corner into the Twilight Zone, everything becomes darker. It's mm-hmm. lower light levels, more blue light. And the temperature of the water in the tanks is a lot lower too. So as long as we keep the light down and the temperature low, the fish are perfectly happy after we decompress them using the decompression chamber that we use. And
they're not stressed out because of the extreme light or extreme, and the cold keeps them, well, the cold is what they're used to. And so they're in a normal, quote unquote, normal environment for themselves.
Right.
Now, with this puncturing thing though, if you puncture the air bladder on a fish, does it heal? Or has it always been, then after that, the fish has always got and has difficulty adjusting to changes in the water depth?
For the most part, they have difficulties for a few weeks, but then it heals. If they don't get infection. So the most common thing is that they die of an infection a few days after, because the needle goes through the body cavity and everything, and sometimes they hit the liver, and it's very hard to do it properly. That's why you opt to do it with a decompression chamber. If you Google fish decompression chamber, there's all kinds. We published a paper about this too, so there's all kinds of articles about it. I think there's a little video about it somewhere too.
So shall we take a couple more questions? How are you? Or just a couple more, and then we'll let you go so that you can have the rest of the evening at sea. Okay. Is there something that you see that regulars? Did you ever let regular citizens on these trips?
Ah,
Joanne Kearns is asking, and if so, how would one volunteer?
right, right. Depending on the trip, sometimes there's a few Academy volunteers that go, but to help with the post-processing. So after we come up with a fish, they help photograph and they help catalog everything. For the diving, it's so training intensive and so risky that we avoid taking anybody that is not part of the team that we dive with constantly. So there's opportunities if you are a volunteer at the Academy. Not very often, but there are, yes.
And I suppose one last, let's see. Is there any way, Yafi is asking, is there any way to ban using fishing lines? Because of the numerous fishing lines.
Right, right. No.
I mean, nets are used.
Nets are used, but they are almost as damaging. I mean, they don't attach to the fish in the same way, but a lot of nets are lost.
Oh,
Let's see, when they go close to a rock and they get attached to the rock, and then they keep fishing. Fish get tangled to them, and they die because they get stuck and they can't get out. And the fishing line is the main way that people catch fish. So banning fishing line would be banning fishing, basically. We could work on producing line that's more biodegradable, that dissolves, yeah. I know that the hooks, there's some that are more biodegradable now than they used to be before. But the only way to ban fishing lines is by creating marine protected areas. So it's closing an area to fishing.
Right.
Yeah, then the lines stop going in, but the fish still come out. Some fish will swim out and then they'll get caught somewhere.
Well, I think we'll let you go here. I mean, there are many more questions, obviously, but we can't answer all of them. We've tried our best to get through the main ones.
Yeah.
And, I, on behalf of everyone who's attending and on behalf of the board of the Friends, wish to thank you for agreeing to give us this wondrous talk on the coral reefs and the good, the bad, the beautiful,
right,
and the ugly, everything that's there. We got an idea of, and so I thank you so much. And you folks go where no man has gone before, just like Star Trek. Honest to goodness, it's like a world onto its own, and exploration of a completely different nature than has ever taken place before. So, Luiz, thank you so much on behalf of everyone.
Thank you. Thanks for having me. And if there's any follow-up questions, I'm very active on social media, especially on Twitter, Coral Reef Fish. So ask me there and I'll answer away.
All right. Thank you.
Thank you.
So much. Have a great evening.
You too.
All right, folks, we finished for today. Stay tuned for next month's Bat program. Batman and Robin, the little guys, are going to be with us. And the month after, the owls are going to be with us. So I hope you enjoyed this talk. I think it was very informative and interesting. And it has been recorded and will be on the website as soon as our IT team gets going, and that's usually on a weekend.